All right. Good afternoon, everybody. My name's Ben Korman, and I am a rheumatologist here at Northwestern. I'm the, newest addition to the Scleroderma program here. And I am interested in sort of a lot of things in scleroderma, but one of my interests that's longstanding is genetics and genomics. I spent a year at the NIH working in one of the premier genomic labs, and so today I'm going to be talking to you about genetics and scleroderma. So first I'm going to give you sort of a general overview of genetics and genomics and what that even means. Then I'll talk a little bit about the specific genetic lesions that we see in scleroderma and how those are relevant. And then I'm going to talk a little bit about why it matters and why you might care about it, and how it might impact things going forward. It's really great to see such a big turnout today. I thank the Scleroderma Foundation and Mary Carnes, John Varga, and the whole team for having me talk today. So just to give you a little bit of background, the whole concept of genetics is that we are born with a set of code, these four bases, A, C, T, and G, that Watson and Crick characterized way back in 1953. So this is what you've heard of as DNA, and is basically the universal code that defines human genes, which basically enables us to turn out the way that we do to a large extent. And so there's a big gap between just seeing that there are these physical structures that they were able to characterize with X-rays to then being able to go and understand what exactly it means and how it can contribute to health and likewise to disease. So it wasn't actually until 1979 that they were first able to characterize a specific genetic lesion that led to a disease, that was thalassemia. And since then, there have actually been thousands of these diseases that have been identified where there's a specific single lesion, so what's called a mutation, which is an error in the genome, something that an individual has that they're not supposed to, that leads to a clinical disease. And so this was thought to be important enough that thousands of people and millions of dollars went into them actually going and decoding all of the 3 billion base pairs that everybody has to actually understand the human genome. And so that was the Human Genome Project, which was completed in 2003. And so now for the last 10 years, we've actually understood what the genetic basis of DNA in human beings is. And so as I mentioned, there are about 3 billion base pairs. So this is one of those four residues, either A, C, T, or G in the DNA. And believe it or not, greater than 99.9% of everybody in this room has the same base pair at the same location in the vast, vast majority of places in the genome, and that's what makes us all human. In fact, 99.1% of our genome is identical to monkey genome. So there's a lot of conserved genome for the whole idea that we need to have things that keep us healthy. And so why do we have all of these pairs of DNA? Well, it's to encode genes. And so genes are basically molecular products that turn into, when they're transcribed, proteins that enable our bodies to function normally. So even though it's only less than 0.1% of our DNA that's different, there's actually still a lot of difference that we can find in the human genome. So there's this idea of what's called polymorphism, or locations within the genome where you can have different kinds of variation. So one individual will have an A and another individual will have a T at the same location. And it turns out there's more than 2 million places in the genome where greater than 1% of humans actually have differences in their genome. And if you look at rarer locations, there's even more places where that's the case. And so, as I mentioned, there are, in doing sequencing of individuals with genetic diseases, things that are inherited, things where you see that the parent has a disease and then the child has a high probability of developing the disease, we've actually identified more than 100,000 individual genetic mutations in 3,700 of those 33,000 genes in the genome, which actually correspond with developing a human disease. And so that's probably just the beginning. Based on sort of mathematical modeling, it's thought that there are more than 15,000 of the 33,000 genes that probably cause human disease. So the long-held dogma in genetics has been the idea of Mendelian inheritance, and this is something you might have learned about back in your high school biology class. This is basically the idea that you can get a gene in one of two different ways, or actually one of three different ways, where basically the gene can either be inherited in a dominant fashion, where basically if you receive one copy, so if one of your parents has a copy of a genetic lesion, then you are basically going to have a 50% chance of inheriting it because they give you one copy of their two in your genome, and so there's a 50% chance that you're going to get the genetic lesion that they have, or what's called a recessive, where basically you need to get one copy from mom and one copy from dad. And so these are sort of classic diseases that we hear about in medicine, things like cystic fibrosis, sickle cell disease, Huntington's disease. In the news now is the BRCA breast cancer. All of these are sort of what are called Mendelian genetic diseases. So they're inherited in these classical patterns of either dominant or recessive inheritance. And if you know that your parent has the disease, or you have the disease, you can predict what the likelihood of a child is to get the disease. However, this is actually a gross oversimplification of most cases of genetics that we're learning today. So what about complicated diseases like scleroderma? Well, this is where genomics, as opposed to genetics, comes in. So probably the most classic genomic disease is Down syndrome. And so we're all born with 46 chromosomes. That's what's shown up on the screen here is what's called a karyotype. And so these are actually the big globs of DNA that we have if you actually look inside of any one of the cells in your body. And so you have 22 of them that are inherited, one copy from mom, one copy from dad, and then you have an X chromosome and then either an X or a Y that defines whether you're a male or a female. So women have two X chromosomes, males have an X chromosome and a Y chromosome. And so in Down syndrome, what happens sort of in an aberrant or an incorrect way is that you accidentally have 3 copies of the 21st chromosome. So this is called trisomy 21, and you can actually see this grossly when you look at a karyotype, and that leads to the phenotype of particular facial abnormalities, mental retardation, that are classic of Down syndrome. And so this same kind of metagenomic thing happens in other more common diseases like cancer, although it doesn't happen in every cell in the body. So in Down syndrome, every cell in the body has 3 copies of the 21st chromosome. In cancer, if you look only at the cancer cells, you can see on a karyotype similar kinds of abnormalities. However, when we look at a disease like scleroderma, it doesn't look abnormal. You would only have the normal, 22 chromosomes and, 2 X's or an X and a Y. So you wouldn't see an abnormality at this level. So in a complex disease like scleroderma, instead of there being one genetic lesion in one gene that's implicated, or a whole chromosome of genes that's implicated, instead there are multiple different genes that lead to the phenotype of scleroderma. And each of the different genes only contributes a small part to whether or not you're going to get the disease. And if you have any one of them, it doesn't actually mean you're going to get the disease. So in order for somebody to be susceptible to scleroderma, they need to have some genetic predisposition to disease, meaning that they need to have a background where they have some of the genetic variants that can make them likely to develop scleroderma. But in fact, just having that probably isn't sufficient for them to get scleroderma. You also need to have other things happen. So probably environmental factors. So in addition to needing to be a genetically susceptible host, you need something else to trigger you to then go and get disease. That's why usually scleroderma doesn't happen in childhood. It's something that occurs in adulthood because there's some injury or insult that leads the genetically susceptible individual to then go on and develop clinical disease. So this is sort of a model of how this works. This isn't in scleroderma, but another autoimmune disease with kind of a similar idea. And the idea is that, you have susceptibility genes. So you have inborn errors of, in the case of scleroderma, mostly the immune system, that make you susceptible to getting the disease or actually to getting any autoimmune disease. And then you have environmental factors. So in scleroderma, we don't know exactly what those environmental factors are. Some of the thought is that they are viruses that you may be exposed to. Other hypotheses include things like cigarette smoke, which hasn't been as well proven, but also things like silica dust in coal miners has been very well implicated. And these things lead to other changes in the genes, in the genome, called epigenetic changes. And the combination of you having that susceptible background and then being exposed to environmental factors makes it so that you can then go on to be susceptible to developing a disease like scleroderma. So there is some evidence that scleroderma is a genetic disease. So just because your parent has it, you aren't likely to get it, doesn't mean that there isn't a genetic component. That was thought for a long time, is that this isn't a genetic disease. It doesn't follow dominant or recessive inheritance. But there are a bunch of studies that were done back in the 1980s where they looked at families with autoimmune disease, including scleroderma, and they found that if you have family members who have an autoimmune disease, that you too are more likely to develop an autoimmune disease. And probably the best evidence of this comes from studies where they looked at patients who had a twin or a sibling, and they looked at the likelihood that the twin or the sibling was going to go on to develop a disease like scleroderma. And so it turns out that if you have a scleroderma, your sibling is actually 15 times more likely than the general population to develop scleroderma. So that certainly argues that there is a genetic component. But on the other hand, only 2.5% of patient siblings actually develop scleroderma. So it's a huge increased risk, but in terms of an absolute risk, it's actually not that great. So that definitely argues there's a genetic component, but it tells you that there's more things than just genetics that contribute to it. Because if it was a recessive disease, you'd expect that number to be 25%. If it was a dominant disease, you would expect that number to be 50%. So 2.5% is actually a huge percentage when you consider the general population. The risk of getting scleroderma is like 0.001% or 0.0001%. But at the same time, it's still a relatively low risk for a sibling. So that moves me on to sort of the modern era of genomics. And so this is sort of the technology of the last 5 or 7 years that has really led us to understanding the genetics of scleroderma the best. So this is something called GWAS, or Genome-Wide Association Study. And so what this was a study that was done, and actually they've done this now multiple times. There have been 6 or 7 of these studies done in scleroderma now, where they look at thousands of patients with scleroderma. So this initial study in 2011, I believe, had 2,200 scleroderma patients and 2,200 or somewhere around there normal healthy controls. And they actually looked at 300,000 of those polymorphic sites in the genome that I told you about, those what are called SNPs, or single nucleotide polymorphisms. And they looked to see what were the differences between patients with scleroderma and normal healthy controls across all of these 300,000 different sites. And so the way that this graph works is these are actually all the 23 chromosomes that I showed you on the karyotype earlier. And this is basically a scale of how significant an association there is. And so in normal genetic or normal scientific studies, significance would actually be way down here. It's at 0.05. But because we're doing 300,000 tests, you would expect that there would be a lot of false positives if you used such a normal criterion. So we actually set the bar very high, and you need to have a very stringent criteria to say that a genetic lesion is actually associated with disease. And when they did that, they still were able to find a number of different, so in this study it was 6 different regions that were all associated with susceptibility to scleroderma. And so this is what's called a Manhattan plot, because these are supposed to look like tall buildings in Manhattan. It doesn't quite look like that in scleroderma, and some of the other diseases it actually does. But what we see in scleroderma, actually very much like almost every other autoimmune disease, is by far the most significant region is this thing called the HLA region, which is an area on chromosome 6, which is part of the immune system and helps define how patient cells are able to identify self versus non-self materials. And so this is something that's the same region that we see in rheumatoid arthritis, in lupus, in autoimmune thyroid disease. So it's actually not a unique finding in scleroderma. And interestingly, pretty much all of these genes that we found are not unique to scleroderma. So these are all things that are found in other autoimmune diseases. So lupus, for example, has all of these same associations that we see in scleroderma. So the genetics of scleroderma actually have a lot of overlap with the genetics of other autoimmune diseases. And so they continued doing studies like this and other studies with huge numbers of patients. So if you look at some of these studies, there was a recent study where they had almost 9,000 patients and 5,000 controls. So these are, incredibly large studies that they're able to do where they look at large numbers of genes, and we've now identified about 30 genes that are all involved in the pathogenesis of scleroderma. So these are things that are statistically significantly associated with scleroderma. So there are 13 with very strong associations that we call either genome-wide significance because they meet that threshold that I showed you, or that they've been found in multiple different studies, and then another 17 that have been found in at least one study to be at least very strongly associated. So the idea is that this is a polygenic disease. It's not one gene that causes you to get scleroderma. And so, as I mentioned, there's a significant overlap with other autoimmune diseases. These are the same genes that we're finding in diseases like lupus and rheumatoid arthritis. HLA is the biggest risk factor, and it's unclear why individuals with the same genetic background, so with the same alterations in the same genes, might develop one autoimmune disease instead of another autoimmune disease, because oftentimes it's the same susceptibility factors, and so it's probably those environmental or other factors that lead you to develop, which disease, and in fact, even within the disease, what type of disease do you develop? Do you get limited scleroderma or diffuse scleroderma? Is your heart involved? Is your lung involved? Is your GI tract involved? Some of that may have to do with some of the variation that we see, and it may be that our studies just aren't powerful enough yet and that we're not looking at close enough groups of patients, but it also may be that environmental factors have a lot to do with it. This is a diagram that I made recently that just sort of tries to put a lot of the genes that we've found from these studies into the immune system and sort of explain how they might be involved in the pathogenesis. This is probably a little bit busy, but the general idea is that all of these genes affect different cells in the immune system, so T cells and B cells, monocytes, dendritic cells, all these different cells that are all involved in immunity and autoimmunity. And so abnormalities in any one of these things, whether it's the HLA region or any of these other genes, the STAT4, all of these genes, the IL-12, IL-2 pathways, all of these things lead to changes in the immune system, which then sort of are a common pathway to leading to abnormal fibroblast activation and tissue fibrosis that leads to the clinical picture of scleroderma. But this is all sort of hypothetical. We don't have any proof for any of that. So what have we learned? That there are a lot of genetic associations. We've found 30, and if we did more studies and sort of looked like other autoimmune diseases, we're probably going to find that there are hundreds of genes that contribute to at least some degree in scleroderma. that genetics don't explain everything. So if you actually go back to that sibling concordance ratio of 2.5%, the genetics actually only explain about half of that. And so it's probable that environmental factors and other things, epigenetics, are explaining a lot of the rest of what's going on. And that genetics make patients susceptible to getting scleroderma, and probably also to getting different types of scleroderma. But there are still a lot of things we don't understand. All the genes that we're finding are immune genes. We're not finding fibrosis genes, and that's the most typical thing that we see in scleroderma is scarring and fibrosis in the skin, the lung, and other organs. So why aren't we seeing that? Furthermore, we don't really understand how having a gene causes you to get scleroderma or a scleroderma manifestation. We don't really understand the role of the environment, although we know that it's probably there. And we also think that there are probably what are so-called private mutations, where one individual may have a single mutation that actually causes them to get the disease, but because it's one person who has it, it's not the kind of thing that could show up in a study of 2,000 people, and we would actually have to go sequencing that person and compare them to thousands of healthy normals to identify a single genetic lesion that might lead them to have basically a single gene variant that causes scleroderma. So that's sort of my overview of genetics. Another piece of genomics is that in addition to the genes that you are born with, another thing that matters is how the genes are expressed. So does the mutation or the polymorphism that you have lead the gene to be expressed differently? Because that's actually what causes the cells to act differently. And so there's this idea of RNA, which is basically the expression of DNA. It's another molecule in your blood or in your cells. And we can actually do studies to see how different genes are expressed. And so there's this big study called a microarray where you can actually look again at thousands of genes at a time, so 40 or 50,000 genes at a time, and see how they're being expressed. And it's not even just a research tool at this point. In other diseases, so this is an example from the transplantation world, you can actually use these microarray studies to determine if a patient is having a transplantation rejection episode and warrants treatment. So this is actually something that's being clinically useful these days. And so in scleroderma, we're actually beginning to get to this. So one of my colleagues, Dr. Hinchcliffe, who some of you have probably met, is actually doing studies like this, where she is doing skin biopsies from patients and then doing whole genome expression studies. And so she's looking at tens of thousands of genes and different patterns of how the genes are expressed. So this isn't what you're born with, but rather what your genes are doing in your skin. And it turns out that we're able to identify different patterns of gene expression in the skin, and that those different patterns have different meanings. So it's actually a very complicated story, but one of the important takeaway messages is that while there are all of these different clinical pictures that we see, so limited scleroderma, diffuse scleroderma, cancer-associated scleroderma, those aren't the patterns we see when we look at gene expression. There's a different set of gene expression patterns, and those may actually be the things that determine whether somebody's likely to respond to a given drug, and whether they're likely to develop different kinds of organ manifestations. And so this is sort of a technology that's in its relative infancy, and as time goes on, we're going to be able to use things like this diagnostically to help better classify whether a patient has some kind of a variant of the disease and how we might want to go about thinking about treating it. I mentioned drugs, so pharmacogenomics and pharmacogenetics is a relatively new area that looks at the genetic determinants of drug effects. And this is something that's also of interest because pharmacogenetics gives us the possibility of looking at your genome and figuring out whether or not a particular drug is going to work for you. It gives us a sense of if you have particular abnormalities that would make it so that you would metabolize a drug too fast or too slow and would be likely to get side effects from it. And it will also enable us eventually, hopefully, to do things like targeted therapy, so that if we find that you have a particular genetic variant, that we would be able to give you a drug that acts on that particular genetic variant. And they're doing this already in cancer, and in autoimmunity there are the beginnings of this kind of what we call personalized pharmacogenetics, where we're picking patients for a given drug based on their genetics. So this leads me to the idea of personalized medicine. And this is sort of the holy grail for people in the genetics field. So what is personalized medicine? It's the right treatment for the right patient. So here's a quote. Personalized medicine refers to the tailoring of medical treatment to the individual characteristics of each patient, and doesn't literally mean the creation of drugs or services that are unique to the patient, but the ability to classify individuals into subpopulations that differ in their susceptibility to a particular disease or the response to a treatment. Preventative or therapeutic interventions can then be concentrated on those who will benefit, sparing expense and side effects for those who will not. And so this is really what we want to get into, is we want to get into the idea of understanding patients' genetic identity so that we can treat them appropriately, so that we're not using the one-size-fits-all approach, giving everybody the same drug, having some people respond, other people not, some people have toxicity. We want to have the right drug for the right person. And so we want to use this information to treat patients based on their genetic makeup. And so if we do this correctly, personalized medicine should aid in diagnosis and prediction of risk. Which organ systems are you likely to have involved? Which complications do we have to watch out for? Help doctors detect disease earlier on when it's easier to treat, so we know, what to look for, so that we're not getting caught by surprise. Classify the disease better, like I explained with the microarray studies. Treat patients with drugs that they're going to respond to, not treat patients with drugs that they're unlikely to respond to or have an adverse reaction to, and furthermore identify new molecular targets that will enable us to identify therapies that are going to work for subsets of patients with unique kinds of scleroderma and other disease. So the problem is scleroderma is a very complicated disease, and that's a good thing and a bad thing. So from a genetic and personalized medicine standpoint, having all of these different types of scleroderma means that it may not be a one-size-fits-all disease. It may be that we have 10 or 20 different diseases, and as we understand it better, as we understand the antibodies, as we understand the organ involvement, as we understand the molecular subtypes and the genetics, putting all that together may mean that we can classify patients as having a subtype of scleroderma and therefore a personalized therapy, so we know what to do with each individual patient. So the path to personalized medicine is probably still years away at this point. And the idea is that right now we're still working on all of these things on the outside, putting in the information technology and the funding just to be able to understand all of this stuff. But then once we get it to the point that we can actually have actionable events, that if you have a particular genetic variant, that we know what to do with it, then we're going to have to counsel patients, we're going to have to educate doctors so that they understand what to do with it, and we're going to have to tell patients what that means and how they want to interpret that information. So there are multiple pieces to personalized medicine. Hopefully it's going to help us in making the correct diagnosis and understanding the disease, but it's going to mean that we're going to need to do a lot of counseling. We're going to have to explain what these things mean, and it's very complicated. You may get a report that's going to be pages long, and you need to be pretty well educated in genetics to understand what it means. Hopefully it'll, as I mentioned, help with pharmacogenetics, help us with prevention, doing screening so that we catch things before they happen or early in their disease course. And the other important thing is that it's got to be about the patient-doctor relationship, that if the patient doesn't want to know something, that you don't tell them, and that you sort of tailor the information that you have to what's actionable. So sometimes you can be very aggressive with these things, and there's a lot of decision making, and it's oftentimes a personalized decision, which is why it's personalized medicine. So it's difficult to interpret all of this, and a lot of doctors don't really know how to interpret genetic results. We're able very easily to look at x-rays and labs, and when it comes to genetics, we often don't understand. And the reason for this is that there's too much information. This is a great slide that I stole from somebody at the NIH, actually the director of the NIH, Francis Collins. And it sort of shows you that human cognitive capacity is mostly the stuff that we normally use in clinical medicine. So things like, what does the patient look like? What do the lab tests show? What does an X-ray show? But then as we start adding all of these tens of thousands of variables that genetic medicine throws in, so the structural genetics that I was talking about, these 300,000 SNPs, add upon that the functional genetics, the microarrays, and the tens of thousands of genes that are differentially expressed. And I didn't even get into proteomics and other kinds of omic studies. You're talking about millions, if not billions, of pieces of information that we're going to be able to understand at a genetic level, and that are all going to have implications. But it's going to be really hard for us to understand what all of this means. And so we actually need computer algorithms and other things that are going to help us so that we can interpret all this information and tell you what it actually means for you. Because just because we can find out what your genetic variant is doesn't mean we can tell you what it means at this point. And so we still need a lot of understanding of what we're finding before we can turn it into something that makes you want to change what you're doing. So there are a lot of challenges, as I mentioned. Current tests that we have aren't that useful. It doesn't classify things as well as we want. People don't understand what the tests mean. We have companies that are coming and trying to sell you your whole genome. There's 23andMe that you may have heard of that's trying to capitalize on this, but doesn't really have a good reason why you would do it. There's issues of privacy. What if your insurance company gets a hold of your genome? What if a hacker steals your genome? They can predict what diseases you're likely to get. So that's a problem, and it's expensive. Another issue is that even though we know what your genes are, just because you have a given gene doesn't mean you're going to get a given disease. And that whole idea of environmental factors, multiple different genes all leading to the same thing or to different things, these complicate our ability to interpret this. And then there's also the idea that all these studies that we're doing in the lab all have implications for the population, for all of the patients in the whole country or in the whole world. But for one person, having a particular thing doesn't mean the same thing as it being strongly associated with the disease in the population. So in conclusion, genetics and genomics are very complicated, and it turns out they're getting even more complicated every day. Every patient is genetically unique, and new technology is going to make it easier for us to understand why people are likely to get a particular kind of disease and why they might respond to a particular kind of therapy. So while we've learned a lot about the genetics of scleroderma, we have more than 30 genes that we can identify as being associated with scleroderma, there's still a lot to learn, but if we do, hopefully it can lead us towards personalized medicine. Thank you.